for human-caused events that result in more than 1 million American casualties, as SARS-2 has, and require general liability insurance to cover it. That would induce the market to price in the cost of negative externalities and cause professional insurance risk analysts to perform those cost-benefit analyses. Now, I'm optimistic about this issue because we just need to buy time. If we can keep pandemic-capable viruses out of the hands of terrorists for a decade, then we can deploy new general-purpose defensive technologies. These range from ubiquitous sequencing that can detect any emerging threat, to perfect protective equipment for our essential workers, to low-wavelength germicidal lights. And these together could protect us from all pandemics, whether natural, accidental, or deliberate. Pandemic proliferation is a solvable national security problem, but only if we recognize it as one. Thank you.
Thank you, Dr. Esfeld. We'll start with Senator Scott from Florida.
All right. Thank you, Chair. Dr. Esfeld, in your testimony, you talk about USAID funding gain-of-function experiments through Deep VZN, the program which specifically conducts experiments geared toward pandemics in virology, and Stop Spillover, which, as you know, researches spillover between animals and humans. Can you talk about what these programs specifically are and why they may be dangerous?
Deep Vision and Stop Spillover are extensions of USAID's long-running PREDICT program, the goal of which was to predict pandemics. That is, to identify viruses in the wild that had a good chance of spilling over and causing a pandemic in humans. And this is part of the laudable One Health program, which seeks to identify essentially hotspots where viruses are likely to spill over into humans and cause a pandemic. Idea is if we find these hotspots, educate the community, teach them what to do in the event of an outbreak, then we might be able to stop it before it reaches our shores. That makes sense. But again, they don't seem to have thought of the security issues associated with publishing a list of pandemic-capable viruses by threat order. Now, we can't necessarily know whether a given pandemic would take off until it's spreading in humans, but there's a narrow set of laboratory experiments that can tell us, does it look like a human endemic virus in certain traits? And these are a tiny subset of all experiments that really aren't very useful for anything else. They don't help with therapeutic development. So part of PREDICT was to take samples of these viruses, bring them back to the lab, run these kinds of experiments, sequence the genomes, share them. They didn't find anything particularly scary, but they found some candidates that looked fairly nasty, including at the Wuhan Institute of Virology. And it's hard to know what USAID did and did not approve, but they are listed as an acknowledgement, as is NIH, on a paper that recombined those dangerous-looking but definitely not pandemic-capable viruses, and then performed experiments to see, did they look like they could plausibly cause pandemics?
So do you think these programs are dangerous?
I think any program attempting to identify an agent that would be widely accessible and could be deliberately released to kill millions of people is pretty much the definition of dangerous, yes.
Okay. Do you think the USAID, whose main job is to provide humanitarian aid globally, has the oversight for programs and experiments like Stop Spillover and Deep VZN, which are not humanitarian in nature?
I think there's a very strong humanitarian case for preventing pandemics. I think that the absence of security oversight means that USAID was probably just not aware of the security consequences of their work. And it remains to be seen whether they will decide that it is inadvisable to maintain a ranked order list of the most threatening viruses.
So do you think they have the oversight ability to handle this job?
It's unclear exactly who they're seeking advice from. My understanding is that they are seeking advice from folks with greater security expertise. And the real question is what actions are going to come of that.
Okay. So would these programs go through a P3CO review?
My understanding is that federally funded research does go through P3CO review. However, it is unclear whether the basic find the pathogens program would go through such review, because until you find it and at least run some characterization to determine whether or not it looks like a pandemic virus, it would not necessarily be regulated. And as previously mentioned, due to the transparency issues with that committee, it's very much unclear what their remit is and is not.
Okay. Do you know who's on the panel for P3 COs?
I do not.
Why wouldn't, is it not public?
My understanding is it is not public.
Why wouldn't it be public?
That is an excellent question.
So do any of the witnesses know why it wouldn't be public?
No.
Is it part of
I know it isn't public, and I don't know why it is not.
So it's part of our federal government, right?
Correct.
And so what do they think Americans are not smart enough to understand it?
You'll have to ask the people at NIH.
Do you know how they made the decision not to make the names public?
No.
Okay. So for each of you, do you think that the P3CO review is comprehensive enough on NIH grants, or do you think gain of function grants have been approved without a P3CO review?
Let's go to Dr. Ebright. I want to leave him out of it, and we'll go through each of you. Dr. Ebright, would you like to respond to that?
Yes. So, as I mentioned in my summary statement, there have been only 3 P3CO reviews in the 4.5 years that the P3CO framework has been in effect. The majority of gain-of-function research of concern, enhanced potential pandemic pathogen research supported by NIH has not undergone P3CO review. It has not undergone P3CO review for the simple reason that the NIH has not identified and flagged the proposals as subject to P3CO review and has not forwarded the proposals for P3CO review.
When we ask the other two to respond as well.
Yeah, I think just echoing Dr. Ebright, it's been a failure, I think, at this point in time, and so we need to find an alternative, which is perhaps to take it out of the NIH, make the oversight outside of the agency that's funding.
One major problem is that gain of function is a terrible term. It applies to most of biotechnology in the raw, and you can try to add qualifiers as you want. But it also inherently does not catch efforts to identify perfectly natural but nevertheless highly lethal pandemic-capable viruses. And it really doesn't matter where the thing comes from. What matters is, do you know that there is a good chance that it causes a pandemic? And again, maybe you don't think we can ever be confident more than, say, 50% for a given virus. But if you get a list of 8 viruses that you're 50% confident, it's possible to make all 8, let them go, and you have got pretty good odds there. So I am concerned by efforts to continue to focus on gain of function because it is so ill-defined, and it seems more productive to narrow in on the classes of experiments that can substantially increase our confidence that a virus is pandemic capable, wherever it comes from. And I certainly echo the calls for external security oversight.
Do you think there's appropriate oversight of existing research after it's been approved to ensure continuous compliance? Or do we just don't even know?
Importantly, the P3CO framework does not mandate compliance. If the P3CO committee makes a decision that the research may not proceed, that decision is only advisory to the funding agency. It is not mandated for the funding agency. The funding agency is free to accept or not accept the decision and is free to determine whether to monitor or not to monitor the progress of the work. This is a major shortcoming.
Thank you.
I just want to interject on the definition, whether gain of function is a good definition or not. That began with the NIH. They gave us a definition, and we started with that. And so I do think Dr. Esfeld's making some good points that we ought to be concerned with viruses that are not created, but that actually come from nature that could cause pandemics. So I think that's part of this discussion, is to try to figure out where we get to. Senator Johnson.
Hey, Mr. Chairman. How long have we had gain of function capabilities? Is that with the CRISPR technology? Mr. Esfeld.
I should probably defer to Dr. Ebright on that.
Dr. Ebright, how long have we even had this capability?
The discussions have been underway since 2002 and 2003. The first examples involved reconstruction of previously eradicated or extinct pathogens. those presented a prototype for understanding experiments that would create new health threats and the need to address them. And again, we're talking a 2-decade-long discussion.
But again, the technology emerged, or they started discussing and then developed the technology? Which came first?
The discussions occurred as the technology emerged. It became possible to do this effectively starting at the beginning of the millennium. The technologies have increased in sophistication and have increased in ease and decreased in cost over time.
Yes, talk about the ease and the cost, because I've heard it is very accessible now, and it's very cheap, and you can basically, a knowledgeable individual can basically do this in their garage.
I think that's an exaggeration, but as Dr. Esvelt has pointed out, given the genome sequence of a virus, it is typically possible to reconstruct infectious particles of the virus, and to do so for costs well under $10,000 US in 1 person month or 2 person months. So for an equipped laboratory, the kind of laboratory that would be present in any state program, and that is present in many research laboratories at academic institutions, this is eminently possible.
So again, reconstructing a virus is one thing, but my understanding what at least theory might be with SARS-CoV-2 is there's gene splicing that occurred here, and some very unusual markers in this furin cleavage site, and beyond my comprehension exactly what that means, but talk to me a little bit about the whole gene splicing aspect of this.
So there's 2 ways to edit a virus. Nowadays, the easiest way is usually to assemble it from scratch using synthetic DNA. But if it's large, then in some cases, it's better to create the altered piece that you wish to insert into the virus, and then use a tool such as CRISPR to do the insertion into the backbone. With respect to the cost, the first virus with a chemically synthesized genome from synthetic DNA was made in 2002. Since then, the cost of gene synthesis has fallen by roughly 1,000-fold. So today, the cost of ordering the components of an infectious influenza virus, for example, the synthetic DNA costs less than $1,000. And that does not require any further editing. That just requires following the reverse genetics protocol, transfecting it into the cells to get the infectious virus. And I estimate that there are around 30,000 people who can do that who have doctorates. And you can say 125 virology PhDs per year in the United States. That's roughly 1/3 in the world. There's probably 4 times as many people who have degrees in other disciplines, such as mine, who can do it. Assume a 20-year career, and that's 30,000 people, add a few technicians.
So
Was there a specific incidence or something that concerned people that caused the pause?
yes, there was experiments in influenza in the Netherlands and Wisconsin that took a virus that was 90% lethal but not airborne, and made it airborne through passage in the laboratory.
And that occurred when?
2013, 2012.
And that caught the attention of who? I mean, who was alarmed by that and instituted the pause? I know it occurred under President Obama, but which member of our health agencies that?
I think Dr. Ebright would be the best to answer that.
Dr. Ebright.
So the possible impetus for the pause was a series of events, laboratory accidents at federal laboratories that have access to and storage of potential pandemic pathogens. The accidents included an anthrax incident at the CDC, another anthrax incident at a U.S. Army facility at Dugway in Utah, and the finding of unsecured vials labeled smallpox virus in an FDA NIH freezer in Maryland. And those 3 incidents occurring in close succession resulted in a hearing in the House Energy and Commerce Committee, and then action by the Office of Science, Technology, and Policy. So the pause was driven ultimately from the White House, from the Obama Office of Science, Technology, and Policy.
So listening to your testimony, I'm assuming all three of you would agree with this statement that this research, and I would say even the mining of dangerous potential pathogens, go crawl in a bat cave to try and pull these things out and bring them into a lab. There's certainly no benefit that overrides the risk. We shouldn't be doing this at all.
Yeah, I call it gain of function and gain of opportunity where you bring a virus back. And as I said, my analysis is that it hasn't contributed to the response to this pandemic.
So we shouldn't do it. I mean, we shouldn't be, we can talk about controls, but bottom line, we shouldn't have controls because we shouldn't even do it. Is that your position as well?
For balancing the potential benefits of prevention against the risk of accidents, it can go either way depending on the numbers you use for those. You can reasonably come out with either answer. When you add the misuse case, that is what absolutely blows it out of the water.
Dr. Ebright?
I believe a strong case can be made, or a case can be made, that certain components of gain-of-function research of concern, particularly components involving pathogens that are currently in human populations, are categorically separate and more justifiable than other components of gain-of-function research of concern. For example, currently SARS-CoV-2, the virus responsible for COVID, is present in millions of humans and is generating variant after variant. Gain-of-function research of concern on SARS-CoV-2 involving the creation of new variants and analysis of the threat posed by them arguably can be justified because this is not creating new health threats that won't arise without intervention, but is addressing a health threat that's in place currently. For that reason, and for reasons like that, I believe enhancing the oversight of the research is a more effective and more prudent strategy than simply banning it.
I would say improve oversight, but would you also agree dramatically limit it?
Absolutely.
Okay, thank you, Mr. Chairman.
Senator Marshall.
Well, thank you, Mr. Chairman, and I hope America is listening today. And to our witnesses, let me say welcome, and I regret that none of you were able to get into the Kansas State University biochemistry program, but I certainly appreciate your credentials that are all here today. I think it's important to not only identify the true problem, but also talk about where we've been. And you all can help us fill into some of the pieces here when we talk about gain-of-function research. It was late in 2011 when the NSABB, which is the NIH's advisory board, stopped two scientists from publishing an influenza gain-of-function study that I believe Dr. Ebright was referring to. And they stopped it because they were afraid it could educate bioterrorists. So this is 2011. Over a decade ago, scientists had figured out how to make H5N1, which is highly pathogenic avian influenza, more contagious. In 2012, those two scientists and 39 others implemented a voluntary gain-of-function research pause on influenza experiments. In early 2012, Dr. Fauci encouraged all influenza scientists to pause gain-of-function and said, and I'm quoting Dr. Fauci, 2012, "It's essential we respect the concern of the public domestically or globally and not ask them to take the word of the influenza scientists." It's interesting to me that Dr. Fauci was focused on the messaging, but he still wanted to continue the gain-of-function research. Again in 2012, Dr. Fauci also said, almost prophetically, that he worried about unregulated laboratories, perhaps outside of the United States, doing work sloppily and leading to an inadvertent pandemic. And he went on to say the accidental release is what the world is really worried about. I go forward to 2014 now, after biosecurity accidents in the United States research labs, which our witnesses have talked about, the Obama White House implemented the second gain-of-function moratorium on influenza plus MERS and SARS because of the potential risk of lab accidents and inherent gain-of-function danger. But gain-of-function still continued at the University of North Carolina. Research later that we shared with Dr. Shi, the bat lady. Nevertheless, clearly the U.S. government and Dr. Fauci knew that the viral gain-of-function research was very concerning. And almost counterintuitively, while Dr. Fauci encouraged United States scientists to pause their GOF studies, Dr. Fauci offshored the pause research to China, not once, but twice. In 2012, Dr. Fauci gave a new grant to Peter Daszak's EcoHealth Alliance for influenza research in China. And then again in 2014, Dr. Fauci gave another grant to Daszak for SARS research in China. Daszak partnered with who? The Wuhan Institute of Virology. In late 2017, the NIH announced a lift on the gain-of-function moratorium, what became known as the P3CO framework, which we referred to, apparently without consultation from a Senate-confirmed State Department head or national security leadership. Also significant, there was no OSTP director in place and only an acting HHS secretary at the helm. So what was the result of this? NIH essentially lifts the moratorium on their own by slipping it in between administrations and self-policing. And today, we can't see the research record for Dr. Fauci's offshore projects because the Chinese Communist Party supposedly has EcoHealth records and NIH resists sharing theirs. So I'll get to my question now. Dr. Ebright, could EcoHealth's research in China have led to the COVID-19 pandemic and Dr. Fauci's worst fears that a lab accident in a foreign lab became reality?
Yes. Lapses in U.S. oversight of gain-of-function research of concern may have caused the current pandemic and could cause future pandemics. The U.S. government funded high-risk gain-of-function research and high-risk enhanced potential pandemic pathogens research at the Wuhan Institute of Virology in 2016 to 2019. The research overlapped the pause that was in effect in 2014 to 2017 and met the criteria to be paused, but was not paused. The research also overlapped the subsequent policy, the P3CO framework that has been in effect from 2018 to the present, and met the criteria for federal risk-benefit review under the P3CO framework, but did not undergo federal risk-benefit review under the P3CO framework.
Thank you so much. I have to stop and point out, too, that USAID, who is knee-deep in this type of research, is part of the State Department where they can get the security advice that they should have asked for before they cleared this with P3CO. Certainly, I believe that this virus came from Wuhan, China, and that it is a product of gain-of-function research. This is a bipartisan national security issue, like several of our witnesses have testified, that this viral gain-of-function could become, and has become, a weapon of mass destruction, that this model, this is a 3D model, what the COVID virus looks like, and this is the gain of function. This is the protein spike, the 2 units that allows this key to fit into the door perfectly, and the cleavage site, and all that. This became a nuclear hand grenade, is what happened. Dr. Quay, then Dr. Esfeld, considering the extreme risk of this research and the incredulous obstruction by the NIH, USAID, EcoHealth in China, should Congress immediately pause this dangerous research?
I think that's an appropriate step for Congress to take.
Okay. Dr. Esfeld?
I think it would be somewhat dangerous to attempt to pause gain-of-function research when it's evident that that term is so malleable as to be evaded at will, and also could plausibly do damage by applying to science that is not specifically directed at potential pandemic pathogens.
Are there any countries that you would say we shouldn't be doing this type of research with?
When it comes to identifying pandemic-capable viruses that could kill millions of people and will necessarily be shared with scientists worldwide who will be able to access them, I do not think that we should be doing it. I do not think that China should be doing it. I do not think that anyone should be doing it, because it is expected to kill 100 times as many people as it might save, even if we could perfectly prevent an identified natural virus from spilling over.
Thank you, Mr. Chairman. I have some more questions if we have time for later, but I yield the floor back. Thank you.
Senator Hawley.
Thank you, Mr. Chairman. Thanks to the witnesses for being here. Dr. Quay, if I could just start with you. You said in your written testimony that the genome of COVID has some of the hallmarks of gain-of-function research, and in particular, 3 genomic regions you say have the signature of synthetic biology. One region has features of the 2 types of forbidden gain-of-function research that are associated with bioweapons development. And you said in your opening remarks that you believe COVID-19 was the product of gain-of-function research and was from a lab leak from the Wuhan Institute of Virology. My question, I guess, is, do you think that China engaged in a cover-up to prevent the world from knowing the true origins of this virus and the lab leak?
I think there's abundant evidence that they have not shared all the information they had at the time. They continue to not share information. I could give you a laundry list of 20 things that they've done, starting with a website with 21,000 viruses on September 12th. 2 a.m., someone was in the Wuhan Institute of Virology. That had been available to virologists for a decade. It was taken offline. It's not been returned. We've asked to see it. No one that I know of has ever seen it. It goes on from there.
Are you concerned with the continuation and expansion of Chinese gain-of-function research?
Well, I think I testified here that in December 2019, they were doing synthetic biology on a cloning vector of the Nipah virus, which is 60% lethal. We just experienced a 1% lethal virus. My estimates would be that could set us back a millennium. The Black Plague was a 20% lethal event, and it was 250 years for civilization to return.
Let me ask you this. How safe were the testing conditions at Wuhan, to your knowledge?
Well, I think that a lot of the Western virologists